All right, today we are diving deep into one of the most incredible molecules in the entire universe. I'm talking about the blueprint of life itself, DNA. We're going to break it down piece by piece and really figure out how this simple, elegant code can build literally everything we see around us. So let's just jump right in with the huge question. Have you ever wondered what the secret code is that builds every single living thing on Earth? I mean everything, from a tiny little bacterium to a massive blue whale, and yeah, that includes you and me. Well, the answer to that question is exactly why we're here. That secret code is deoxyribonucleic acid. You probably know it as DNA. You can think of it as the ultimate instruction manual for all of life. And it's famous for that beautiful, super efficient shape, the double-stranded, twisted helix. Okay, so how in the world does this amazing molecule actually work? Well, to really get it, we need to zoom way in and look at its most fundamental parts. So let's start with the absolute basic building blocks of this incredible code. All right, the single most important unit we need to talk about is the nucleotide. If DNA is this giant instruction manual for life, right, then a nucleotide is like a single letter in that book. Every single word, every sentence, every instruction, it's all written using these fundamental pieces. So every single one of these nucleotides has three distinct components. First up, you've got a sugar, and it's called deoxyribose. Then there's a phosphate group. Now these two parts, the sugar and the phosphate, link up over and over again to form the long backbone of the DNA strand. But the third part, well, that's the star of the show, a nitrogenous base. This is where the actual information, the code itself, is stored. Yeah, this nitrogenous base is where the genetic code really lives, and there are 4 different types. You've got adenine, guanine, cytosine, and thymine. Let's just call them A, G, C, and T to make it easy. Now, they're not all the same shape. They fall into 2 little families. The purines, which are A and G, have a double ring structure. And the pyrimidines, C and T, have a simpler single ring. And this shape difference is super important, as you'll see in a second. And right here, this is it, the absolute golden rule of DNA. These bases don't just pair up randomly. Nope, there is a strict, unbreakable law. A always pairs with T, always, and C always pairs with G. This is what we call complementary base pairing, and it is the entire secret to how DNA holds its famous shape and, crucially, how it copies itself almost perfectly. So what holds these pairs together? Well, they're connected by something called hydrogen bonds. Now here's a cool detail. The AT pair is connected by 2 of these bonds. But the CG pair, it uses 3. So what does that mean? It means the CG connection is way stronger. It might seem like a small thing, but it has huge implications for everything from how DNA gets packed into a cell to what temperature you need to unwind it in a lab. All right, so we've got our nucleotides, our bases, and our pairing rules. We have all the individual pieces. Now let's zoom back out and see how they all come together to build probably the most iconic shape in all of science, the double helix. So the classic shape of DNA you always see is what's called the B form. It's a right-handed helix. But here's a really key feature. Its 2 strands are anti-parallel. Think of it like a two-way street. One strand runs in one direction, what scientists call the 5' to 3' direction, while the other strand runs in the exact opposite direction, 3' to 5'. This setup is absolutely critical for how our cells read and copy the DNA code. And you know, this structure is incredibly precise. It's not just some random twisty thing. It has a very specific, very regular geometry. Get this. One complete 360-degree turn of the helix measures exactly 3.4 nanometers. And packed inside that single turn, you'll find exactly 10 base pairs, kind of like 10 rungs on this tiny spiral staircase. And finally, the width, or the diameter, of the helix is always a consistent 1.9 nanometers. It's this incredible predictability and order that's the key to storing a huge amount of information so safely and compactly. Now, you might think this twisting creates a perfectly smooth, symmetrical spiral, but it doesn't. Instead, it actually forms 2 different kinds of indentations, or grooves, along the sides. There's a wide, spacious one called the major groove, and then a much tighter, narrower one called the minor groove. And this is such a brilliant example of how structure creates function. I mean, why do these grooves even matter? Well, they are absolutely critical. They basically act as docking sites. Proteins can come in, latch onto these grooves, and get just enough access to the edges of the bases inside to read the genetic sequence. And the best part? They can do it without having to unwind the whole molecule. It's like a clever little access hatch to get to the instructions. Okay, so just when you think you've got DNA all figured out, biology, of course, throws a curveball. That classic B form we've been talking about, turns out it's not the only shape DNA can take. So let's check out some of its surprising variety. So yeah, B form is the standard for sure, but under certain conditions in the cell, DNA can actually change its shape. You've got the A form, which is a bit wider and more squished down, and then you have the really wild one, Z form, which is a left-handed helix. It twists the other way and has this funky zigzag backbone. And these aren't just weird little quirks. These different shapes can actually play a role in controlling which genes get turned on or off. It just goes to show how dynamic this molecule really is. And DNA's variety isn't just about its shape, it's also about where you find it. Most of it, our main genome, is packed into the nucleus of our cells. That's about 3 billion base pairs of linear DNA that we inherit from both of our parents. But get this, we also have a second, totally separate genome. It's hiding out inside our mitochondria. You know, the power plants of the cell. This mitochondrial DNA is tiny in comparison, it's circular, and here's the cool part. You only inherit it from your mother. Okay, we have gone deep. We've seen the parts, the structure, the different forms and locations. But now it's time to zoom all the way back out and ask the biggest question of all. What is this all for? What does DNA actually do? Well, as you can probably guess, its functions are pretty grand. First and foremost, it stores the entire genetic library for an organism. All of it. Second, it holds the blueprints for building proteins, which are the little machines that do pretty much all the work in our cells. Third, it has to be able to replicate itself almost perfectly to pass all that information on to new cells and, of course, to the next generation. And finally, when you get right down to it, it's the ultimate source code that defines the unique identity of every single living thing. So when you boil it all down, DNA is the ultimate source of information for the cell. It's not just a molecule. It's a library, it's a cookbook, it's a hard drive, all rolled into one incredible package, holding every instruction needed to build and run an entire organism. So, if there's one thing to take away from all this, it's this. Everything about you, from the color of your eyes to the way your brain is wired, it all comes down to the specific, unique order of those 4 simple letters. A, T, C, and G. That sequence, that's the code that writes the story of life. And that leaves us with one last, really fascinating thought to chew on. The DNA of any 2 people on this planet is 99.9% identical. So just think about what that really means for a second. All the diversity, all the individuality, all the uniqueness that makes you, you, is written in that tiny, tiny little 0.1%.